Welcome. Today, we're diving into pharmaceutical quality risk management, specifically how it applies to analytical testing. I'm Dr. S.K. Singh, and together we'll explore the strategies and principles that help ensure accuracy and reliability in this critical area. Let's get started. Let's start with an introduction. This is where we set the stage, laying out what we're about to explore together. It's the moment to get curious, open-minded, and ready to dive in. We'll outline the key ideas and why they matter, so you know exactly what to expect as we move forward. Let's take a closer look at the regulatory framework guiding our work. We rely on key ICH guidelines, like Q9 on quality risk management, Q10 covering the pharmaceutical quality system, Q14 for analytical procedure development, and Q2 on validating those procedures. Then, there's the FDA, with its CGMP regulations, data integrity guidance, and quality metrics program. We also follow WHO's GMP guidelines and technical reports, alongside the EU GMP standards, including their pharmaceutical quality system and annexes on qualification and validation. These form the backbone of our quality approach. Let's walk through the quality risk management lifecycle. It starts with risk assessment, where we identify and analyze potential risks. Then comes risk control, focused on reducing or eliminating those risks. Next is risk communication, making sure everyone involved stays informed. Finally, risk review keeps the process alive with continuous monitoring and improvement. This cycle moves from identifying risks to evaluating them, controlling them, and then reviewing, a continuous loop that keeps quality front and center. When we look at potential risks, they fall into several key areas. Personnel risks include things like inadequate training, human error, and incorrect calculations, all factors tied to the people behind the process. Then there are instrument risks, like calibration failure, malfunctions, or software errors that can throw off results. Method risks cover poor robustness, inadequate validation, and uncontrolled changes. Basically, weaknesses in how we do things. We also have data integrity risks, such as unauthorized access, missing audit trails, or data manipulation. Finally, sample risks like contamination, improper storage, or incorrect preparation can all impact outcomes. Each area demands careful attention. When it comes to assessing risk, we rely on a set of proven tools. One of the most popular is FMEA, failure mode and effects analysis, which helps us identify potential failures before they happen. Then there's HACCP, focused on hazards and critical control points, especially in food safety. We also use fishbone diagrams to trace root causes and risk ranking to prioritize what matters most. Fault tree analysis breaks down complex risks logically. Among these, FMEA stands out as the go-to tool in analytical labs. Let's take a closer look at how we assess risk using failure mode and effects analysis, or FMEA. We categorize risk based on the risk priority number, or RPN. An RPN below 50 signals low risk. Between 50 and 100, we call it moderate. Anything above 100 is high risk. We also break down impact scores from 1 to 10. Low impact falls between 1 and 3, medium from 4 to 6, and high impact from 7 to 10. This system helps us prioritize what needs attention first. Let's take a look at an example calculation from an FMEA, failure mode and effects analysis, applied to an HPLC assay method. Here, we're focusing on one specific failure mode, incorrect mobile phase preparation. We score this failure by 3 parameters: severity, occurrence, and detection. Severity is rated at 8, which means the impact is quite serious. Occurrence is 5, so it happens occasionally. Detection is 4, indicating moderate chances of catching it before it causes harm. This kind of breakdown helps us prioritize risks and decide where to focus our improvements. Let's look at how we handle unexpected results. In this case, an assay coming in at 88%, well below the expected range of 95 to 105. When this happens, we kick off a root cause investigation following our out-of-specification procedure. We assess potential risks like instrument failure or errors in standard preparation, both rated high risk. Other factors, like sample degradation or calculation mistakes, carry medium risk. This structured approach helps us pinpoint the issue and keep quality on track. When it comes to managing risk, we've got several layers of control strategies. First, personnel controls, making sure everyone's trained, qualified, and regularly assessed for competency. Then, instrument controls. This includes installation, operational, and performance qualifications, plus preventive maintenance and calibration programs to keep equipment reliable. Method controls focus on robustness studies and managing the method throughout its life cycle. Finally, data integrity controls ensure trustworthiness with audit trails, electronic signatures, and strict access controls. Together, these strategies build a strong defense against risk. Let's look at an example of risk reduction in action. This is where we identify potential problems early and take steps to minimize their impact. It's about being proactive, spotting risks before they become issues. By doing this, we protect our projects, our teams, and our goals. Risk reduction isn't just a safety net. It's a smart strategy that keeps everything moving forward smoothly. Now let's talk about risk-based analytical method validation and how quality risk management plays a key role. We focus on critical method attributes, things like accuracy, precision, specificity, and robustness. These are the qualities that define how well our method performs. Then, we look at critical method parameters, factors like pH, flow rate, column temperature, and mobile phase composition. Our goal here is clear: to identify which parameters have the biggest impact on analytical performance, so we can control what matters most. Data integrity is a major focus worldwide, often flagged during inspections. To manage this risk, we rely on the ALCOA Plus principles, a framework that ensures data is complete, consistent, enduring, and available when needed. Each piece of data must be attributable. We need to know who performed the activity. It has to be legible so records are easy to read. Data should be contemporaneous, recorded at the time of the activity. It must be original, preserving the source data. And finally, accurate, correct, and complete every time. We're seeing a clear impact from effective risk mitigation. Laboratory failures have steadily decreased over the past 3 years. Out-of-specification events dropped from 15 in 2022, down to just 6 so far in 2024. This improvement comes from ongoing risk reviews and continuous improvement efforts. We keep a close eye on triggers like OOS and OOT results, deviations, audit findings, method failures, and regulatory inspections. By tracking these trends, we catch issues early and keep quality on track. Let's wrap up with the key takeaways. Quality risk management, or QRM, is no longer optional. It's a clear regulatory expectation under ICH Q9. Embracing risk-based thinking doesn't just improve our analytical reliability, it directly enhances patient safety. Among the tools we use, FMEA stands out as the most common for lab risk assessment. The Risk Priority Number, or RPN, guides us in focusing our mitigation efforts where they matter most. When done well, QRM cuts down on deviations, out-of-spec results, and data integrity issues. And by weaving QRM into every stage, from method development to routine testing, we build a stronger, more resilient pharmaceutical quality system. That wraps up our presentation. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you. We look forward to continuing the conversation.